The discovery of antibiotics is one of the most important advancements in clinical medicine and public health. It's laid the foundation for a number of other advancements, including the ability to perform surgeries more safely and the reduction of infant and maternal mortality rates. Many antibiotics are derived from either bacteria or fungi. For example, penicillin, which is secreted by the fungus Penicillium, can kill bacteria. This is because microbes use antibiotics to fight off other microbes. But the use of antibiotics, and more broadly antimicrobials, which include medications that target not only bacteria, but also viruses and fungi, has exploded in recent years. Antimicrobials have been used on an industrial scale, partially because of overprescription in humans, but more so because of routine use in farm animals. In fact, a good number of antimicrobials are excreted from humans and animals unchanged, and these get flushed into wastewater, which allows pathogens to be perpetually exposed to antimicrobials. In response to this enormous selective pressure, many pathogens have become highly resistant to antimicrobials. Now, when it comes to bacteria, generally speaking, there are 4 mechanisms for how they become resistant to antimicrobials. The first mechanism is antibiotic inactivation, or modification, which is where bacteria develop specific enzymes that destroy and inactivate antimicrobials. One example is beta-lactamase, which is a bacterial enzyme that destroys antimicrobials that contain a beta-lactam ring, like penicillins and cephalosporins. As a result, bacteria that produce beta-lactamases are immune to the action of many beta-lactam antibiotics. The second mechanism is the alteration of a target or binding site. An antibiotic that can't bind anywhere is rendered useless. One example is a methicillin-resistant Staphylococcus aureus, or MRSA, which modifies its penicillin binding locations, making them unable to be bound by any antibiotics in the penicillin family. The third mechanism is by bypassing metabolic inhibition. For example, some antibiotics, such as sulfonamides, disturb the metabolic pathway that synthesizes folic acid, which is vital to DNA-based organisms. But to get around this, some bacteria are able to scavenge for folic acid from the environment, completely circumventing the action of sulfonamides. The fourth mechanism relies on preventing antibiotic accumulation. One way that bacteria do that is by decreasing the permeability of their membrane to the antibiotic. The other way is to create efflux pumps, which pump the antibiotic out of the bacteria. Both actions result in lower intracellular antibiotic concentrations, which reduces or even negates the effect. Now, these mechanisms work in different organisms against different antibiotics. It's an ongoing challenge. For every new antibiotic that's identified, pathogens rely on selection pressure to find a mutation that somehow allows them to thrive in the presence of that antibiotic. Alright, as a quick recap, bacteria can achieve antibiotic resistance through 4 fundamental mechanisms. Inactivation physically destroys the antibiotics before they can do their job. Alteration of the binding site leaves the antibiotic without a binding site, and therefore unable to work. Alteration of the metabolic pathway completely bypasses the activity of the antibiotic. Finally, reduction of accumulation keeps the intracellular levels of antibiotics low.